My name is Russell Hansen, and I'm currently working at the Dana-Farber Cancer Institute and Harvard Medical School. The Cures Now is working on funding research to help cure diseases and develop new technologies to enable doctors to work with patients in a better way. We're really dedicated towards bringing advanced technologies to the clinic. We believe there is a lot of disconnect between medical devices and the way doctors want to treat patients and what can be done. We believe strongly that the funding that the Cures Now is using can help develop and really get them in the hands of the practitioners in emergency rooms and cancer institutes, everywhere that they can be applied. Something that is very exciting right now is that a number of different technologies for imaging tissues can be observed using new optical and electron microscope methods. This can improve the diagnosis of brain disease like Alzheimer's and determine how well different chemotherapy drugs work in terms of figuring out how blood flow can come to the tumors. It may be that some aspects of Alzheimer's are actually due to something that we can't currently detect. That could be something that we would be able to diagnose either via brain scanning or optical or electron microscope methods. The reason we're so interested in the nematode worm is that we think it would allow us to ramp up simulating a whole human brain further down the line. You have to start somewhere, and we believe the nematode as a simple model organism can help the technology develop to a point where, at some point, it would be possible to test out treatments on a model brain. It's sort of like Craig Venter and coworkers have developed a synthetic organism. What we are interested in doing is developing a synthetic in silico brain that could be used for the basis of molecular therapies and other therapies as well. One of the most compelling reasons we see for understanding the brain through simulation is to allow medicines and treatments to be developed in silico. So instead of terminally ill patients or patients who are seriously ill being given medicines that have not been fully tested or have serious side effects, many of these medicines that are going through the testing phases can be tested and hopefully verified with a high degree of accuracy using simulation or accurate models of brain atlases and brain maps. We're also very interested in gene therapies. We currently have a method that uses synthetic biology to target cancer cells very selectively, and therapeutics that can target specific cancer cells and none of the patient's normal cells. We're using genomic methods to determine under what circumstances using a DNA damaging agent, cisplatin, should be used to treat ovarian and breast cancer patients. We believe this will significantly improve patient care. If we had the necessary funding right now, I'd be very interested in current tissue imaging technologies, particularly for neuronal and synaptic connectivity. In addition, I'd be very interested in new cancer therapies that are based on synthetic biology signatures and expression levels of different genes as hallmarks of different types of cancer from different tissues like kidney or lung or ovarian tissue. Thank you for your attention. If you would like to reach me, you can reach me at russellhansen@thecureisnow.org.